I think we're there. How's everybody doing? Ready for the big exam? The enthusiasm is overwhelming. How's studying going? Omar? Okay. I thought you had a question. There wasn't a question. What should I say? I did put an email out yesterday with a sample exam, which I had meant to do earlier, and I apologize. I hadn't done that. I thought I had it on the web page, but it wasn't there. So I do encourage people to look at that sample exam because it is a different format than you had last term. And as you will see, the exams this term, time will really not be a factor for you. And they are all what I call true-false, which is what those first ones are, and short answer and matching. Those first ones look like they're sort of weird multiple choice, but they're just true-false. It's really all they are. So you circle if it's true, you don't circle if it's false, and if you circle when you should, you get a point. If you don't circle when you should, you get a point. And other two combinations, circling when you shouldn't or not circling when you should, won't get you any points. So that's really what those are. Okay? Yes?
That first section seemed like it was shorter.
Is what?
The first section seemed like it was
Shorter?
Yeah.
Okay. It's a different format, so it's kind of hard.
I meant the other two sections.
So the first section typically has about 20 or so questions, 20 or so points. So yeah, it varies a little bit from exam to exam. Yeah. So each one of the individual ones in there is worth 1 point. Okay. So a total of 4 points for each question. Yeah. Yeah. Yes.
I've had you before, so just curious.
You poor thing. Or your lucky thing, maybe.
Yeah. Like DNP instead of dinitro, whatever it is.
Yes.
Is that okay?
Okay. So a question about since you haven't had me before, it's a good question. So any abbreviations I use in class, you're welcome to use. You have to get them the same, so you can't call it DPN, for example. But as long as you're using abbreviation that I use in class, you're always welcome to do that, yes. Okay, I guess we go home then, right? All right, so what's up?
So when you were talking about the citric acid cycle, you were saying that oxygen was indicative of the presence of NAD and FAD. Was that because of oxygen's role in oxidative phosphorylation and NAD being produced there?
Okay. So his question has to do with the basically the relationship between oxygen, NAD, and FAD, I think is what is a safe way to put that. Okay, so you're exactly right. That is that what you saw was that in electron transport, in order to make NAD, in order for electrons to flow, you had to have oxygen. And in order to have oxygen, in order to have NAD, electrons had to flow. So if you have oxygen, electrons are flowing, then you're going to produce NAD. Same is true of FAD because of the complex 2 relationship and so forth. So absolutely, oxygen is a requirement for NAD. And anytime you have plenty of oxygen, okay, you will have abundant NAD. Yeah. Yes?
Just concerning electron transport, what are the big picture take-home messages for complexes 1, 2, and 4?
1, 2, and 4. Interesting selection of.
3 is Q cycle, 5 is the 8.
So you're asking what's the important things about those? Is that what you're asking? Well, again, anything I talked about with respect to those are important. So.
Like, what's the general? Can you just walk me through it?
Well, okay. So I guess what I would say to that, I'll say a few things in general about it, but I did give a lecture on this, so I'm not going to give the lecture a second time. Complex 1 is obviously an entry point for the electrons from NADH, complex 2 being an entry point for electrons from FADH2. And that difference means that there's more protons that are pumped by electrons coming from from NADH than there are from FADH2, because only electrons from NADH go through complex 1. So that means then that if there's fewer protons pumped, there will be fewer ATPs that are made, because the protons produce the proton gradient, which are what's necessary for the complex 5 to make ATP. So we think of NADH as making more ATPs. And I will tell you that we tend, as I said in class, people get all obsessed counting electrons and protons and so forth and spend their whole careers on it. And it's not really relevant for our purposes. But I will tell you something that hopefully won't confuse you, but that is relevant to this. And that is that we tend to say, well, 3 ATPs per NADH, 2 ATPs per FADH2. All right? And there's no reason that needs to be whole numbers. Okay? They're really not whole numbers. It might be 2.8, it might be 2.6. And this is what some of the things that people argue about. Because not every proton that goes out ends up coming back in. Right? So we can imagine protons going out and making out in the cytoplasm and going and binding to something and so forth. And so they don't necessarily make it back in to the thing. So if we had perfect relationship with that, then probably 3 would be about right for NADH and 2 would be about right for FADH2. But that's not the case. All right? So if we say 3 and we just kind of remember in the back of our heads that it's about 3 and it's about 2, that's fine. And the reason I tell you that is because if you look at different books, you'll see I talk about 38 ATPs per glucose. And 38 ATPs per glucose come about if you make that assumption of 3 and 2. Some books will say 34. Some books will say 32. Some books will actually say 30. All right? And that depends on how many ATPs there are per NADH and FADH2. For our purposes, it really doesn't matter. We get a lot of ATPs produced as a result of electrons coming from NADH and FADH2. You get that message, you've got it. Okay? Complex 4 was the one that you asked about. Complex 4 is, of course, the terminal electron acceptor destination. That's where it's going to combine with oxygen and make water. And I had a very, very good question today. Somebody, I believe in the eCampus, asked me about complex 4. And I'll tell it to you here because I didn't know the answer to it. And so I don't know everything, obviously. But I'd never thought about it before. And the question was, carbon monoxide inhibits complex 4. Is it covalently binding? And that's the way that it's doing that. Okay? Well, that's a very good thought because if we think about what's happening, oxygen, all right, molecular oxygen is making that peroxide bridge inside of the complex 4. I showed you how the electrons are coming on, then the protons come in, and it gets reduced, and water ultimately gets made. So if carbon monoxide is actually blocking complex 4 by getting in that same site, it must be doing some kind of a bond, and that's kind of unusual. I hadn't really thought about it before. So, like I said, it doesn't really matter for this exam, but it is an interesting thing to think about that mechanism of carbon monoxide doing that blocking. Okay. So I hope I've given you some insights there. So, okay. Yeah.
I have a little more specific question. Is the ATP-ADP translocase, is that an antiport even though they're not going at the same time?
Yeah, that's a good question. ATP-ADP translocase, is that an antiport? Absolutely. And in fact, if you look at the sodium-potassium ATPase, for example, they're not going at the same time either. In fact, most of these don't go at the same time, but they go in the same cycle. So one cycle of that will put one out, one in. Or in the case of sodium and potassium, you get three sodiums in and 3 sodiums out and 2 potassiums in, right? So, so that's it doesn't have to equal numbers, but in the same cycle, that same thing will happen.
And on that same thought, is that ATP-ADP translocase, is that both on the inner membrane and on the outer membrane? Because the ATP is on.
Okay. Yeah, so his question is the ATP translocase on both the inner membrane and the outer membrane? As far as I know, it's only on the inner membrane. And I haven't said much about the outer membrane, and I'll tell you about the outer membrane just a little bit. The outer membrane I like to think of as a wet blanket. Okay? So a wet blanket will cover something. It will sort of hold, to some extent, heat or something in. But there's all kinds of things that will leak through a blanket, right? It's not watertight. And that's really the way I envision the outer mitochondrial membrane. I'm told that molecules of up to a size of about 50,000 in molecular weight can pass through it. So it's pretty big. It's got pretty big holes in it and so forth. So that's why I say if we imagine protons, we talk about them going out to the intermembrane space between the outer and the inner membrane. We can imagine that those protons aren't necessarily confined to that region. That's another reason not every proton that goes out comes back in. But a wet blanket will sort of constrain things. You're probably going to have a fair number of protons that are going to be in the intermembrane space. Make sense? Is that it? Okay. Yes, sir. Anything I said in class, right? So whenever the question is, how much do we need to know? It's whatever level I talked about in class, right? Yes?
Could you explain a little bit about pyruvate dehydrogenase complex?
Okay, what did you have in mind?
Just what the cofactors of cofactors.
Okay, so if we look, oh, there we go, wrong thing. At the pyruvate dehydrogenase complex, okay, it is a complicated structure that has, I was looking for, no, that's not it either. Where's it at? Core structure. Okay. It is a complex structure, many subunits that are there, and it is totally dependent upon the 5 coenzymes that I talked about in class. Each coenzyme has a specific role, and each of those coenzymes also is found in the alpha-ketoglutarate dehydrogenase enzyme in the citric acid cycle. And that's not surprising because, again, they're both alpha-keto acids. They both are doing a very similar type of decarboxylation reaction. And they're both producing a molecule that's linked to CoA. In this case, it's linked to acetyl-CoA. It makes acetyl-CoA. In the case of the alpha-ketoglutarate dehydrogenase, it's making succinyl-CoA. Both are oxidative decarboxylations. Now, the steps in the process that I talked about relative to that were not that. They were this. They were not that. They were. Okay, well, I'll eventually get to it. So the first step in the process is the decarboxylation. The decarboxylation produces something that a lot of people refer to as activated acetaldehyde. So activated acetaldehyde basically means it's acetaldehyde, and acetaldehyde, if it were free, would be carbon, carbon, and double bonded oxygen. So it's in a reduced form that's bound to this, but it's reactive, and it has to be bound to TPP. Otherwise, it will react as acetaldehyde. Okay? So this guy is the first product of the reaction. You notice it's been decarboxylated. It started out as a 3-carbon compound, and this looks like it's carbon monoxide, but it's actually CO2 that's released over here. Now, that reaction is the one that I talked about in class. That is different potentially in bacteria and in yeast than it is in humans. This part goes just fine. It's the same in bacteria, yeast, and in humans. However, bacteria and yeast can take that hydroxyethyl TPP, or the also known as the reactive acetaldehyde, activated acetaldehyde, I can't even say it right, the active acetaldehyde, and they can bypass the next step, which is the oxidation. So we've done here a decarboxylation, but no oxidation. That's why this has an OH on it instead of a double bonded oxygen. We had to reduce the double bonded oxygen here to provide the electrons to release the carbon dioxide. Okay? Now, yeast and bacteria can release this, and they release it as ethanol. Okay? So no oxidation doesn't lead to an acetaldehyde. This has been reduced, okay? And now it's going to be released as ultimately as ethanol. All right. We can't do that. We have to transfer this to lipoamide. So for us, the next step is right here. Here's our reactive activated acetaldehyde that's right here, okay? And we combine it with lipoamide, and you notice we start out with a disulfide bond, meaning this is in the oxidized state. We transfer this guy over here, which has electrons, transfers the electrons to reduce that disulfide bond, in the process oxidizing that carbonyl, that hydroxyl group to a carbonyl, to a ketone, and leaving us with this. All right? So this is where the oxidation step happens. That's obligatory for us. It's not obligatory for the bacteria and yeast. All right? Because this is obligatory for us, we can't make ethanol. Okay? At least we can't make ethanol by this reductive mechanism that the Bacteria and yeast can do. Okay? Now, this guy here has to then go through the rest of the cycle to ultimately be transferred to the acetyl-CoA. So this happens in the next step, where coenzyme A is combined here. This is pulled off, linked there. And now we're left with fully reduced lipoamide, okay, dihydrolipoamide. That fully reduced dihydrolipoamide is going to have to be oxidized in order to get back to its original state. Oxidation of it will result in reduction of, first, FAD. Okay? So we see that occurring right here. Here are the electrons coming off of here onto FAD. Protons, of course, come with that. We get FADH2. We're back at the disulfide bond state that we started with there. And then, and as I said, this is an unusual reaction, the electrons from FADH2 are transferred to NAD to make NADH. Now, you might wonder why that doesn't happen in the mitochondrion so that we would get more ATPs. It turns out this is not energetically very favorable, and the only way it really happens here is by the way the electronic structure of this complex is inside the enzyme. That's not really the same as an FADH2 that's floating freely. So they're really at a different state, and because this is actually attached to the enzyme, because it's attached to the enzyme, this reaction is possible. Okay? Well, so what we've done now is we've decarboxylated, we have produced acetyl-CoA, we've ultimately produced NADH, and we're ready to go to the citric acid cycle with the acetyl-CoA, and we're ready to go to the electron transport system with the NADH. This is all happening in the mitochondrion. Okay? It's all happening in the mitochondrion. Pyruvate dehydrogenase is located in the mitochondrion. More than you wanted to know? Yeah. So, okay. So arsenite, which is the compound, will react with the lipoamide and not allow this process to occur. Okay. So if we look at the arsenite, which I thought was on here somewhere. Anybody see it? Where's it at? Structure. Is it the bottom? Oh, there it is. Okay, yeah. I'm reading the first part, not the last part. Okay, so here's the combination of arsenite with the sulfhydryls that are there to make this bridge compound. And if you don't resolve that compound, you are dead. Okay? This can be resolved. Okay? The bigger problem that happens with arsenic poisoning in the world is that there's all kinds of wells in parts of the world that have arsenic in their water. And so the poisoning that's happening there is happening over a period of time. It's not happening as if you had accidentally eaten a bunch of arsenic, in which case it would be quite clear what's happening and quite clear what needed to happen with this. Over a period of time, arsenic poisoning kills a lot of people because they don't even know it's going on as it's going on. Yes, back in the back.
We can refer to that as BAL.
You may refer to that as BAL. That's correct. Yep. Yeah.
Do you have the same transfer of electrons from FADH to NAD and the alpha-ketoglutarate mechanism?
You have exactly the same mechanism going on in the alpha-ketoglutarate dehydrogenase mechanism. The only difference in the two is that pyruvate has 3 carbons, and the bottom one is a methyl. In the case of alpha-ketoglutarate, you have 5 carbons. The bottom one is a carboxyl. But the top 3 are identical. You got carboxyl, ketone, carbon. That's true whether it's a pyruvate or if that's alpha-ketoglutarate. So the top parts of that, which is where all the action's occurring anyway, are identical. Cells are lazy, so rather than completely evolve a new enzyme to handle alpha-ketoglutarate, it makes perfect sense to adapt it, the pyruvate dehydrogenase enzyme, to make that work for alpha-ketoglutarate. Yes, back in the back.
The Monday's lecture.
Yes.
On cholesterol.
Yes.
The hyperlink, it says that the video is reprocessing.
Yes. Yeah. I had several problems with it during the day. I think it should be ready now. Why don't we even take a look at it and see? I think it should be ready now. I had several issues with the video. I apologize it took so long to get up there, but I think it's ready. So let's see if it goes. And survey says, oh, still, no, it's there. Ta-da.
Most common question in everybody's mind is, where does the material for the exam stop?
Oh, shut up, Ahern.
Compounds that ride for cholesterol.
Okay, so yes, it is there finally. So sorry, it took a while. Sometimes the videos there are technical issues with them, and I have to sort of either restart a crashing program, or there's problems with the upload. So the upload process takes about an hour, and then YouTube has to crunch on it after it gets up there. So there's sometimes problems that happen, and that's unfortunately what happened today. So sorry it took so long to get up there, but it is there. Yes. What is ketone body synthesis? Good question. We're going to talk about that after the exam. So it's just an alternate path. For our purposes right now, the only thing you need to know about ketone body synthesis is it's an alternative to cholesterol synthesis. We'll talk about what's happening with ketone body synthesis when we talk about fatty acid oxidation and fatty acid synthesis. And so it's relevant there. But for right now, that's all you need to know about. Yes? Sure, ATP synthase. Yeah, I'd be happy to. Let's see. If I go ATP synthase would be back here. And if I go ATP synthase mechanism, that's a little big. How about ATP synthase mechanism? Yeah, it's a little better. Okay. So this is the ATP synthase. We're seeing that head of the mushroom. And we're seeing this inner part called gamma. Right? So gamma is the rotating part of the mechanism. And gamma is caused to be rotated by the movement of protons through the ATP synthase. So the reason protons are moving is they're going from a high concentration to a low concentration, which means they're going from the intramembrane space down into the matrix of the mitochondria. All right? And there's a gradient that's been there that's been produced by the electron transport system. All right, so we look at this and we see that there are three places that are really, I say identical, but they're very slightly different in shape, and that shape varies only as this guy rotates. All right? So in terms of the amino acid sequence, they're all identical. All right? They're all identical, but their shape changes as this guy rotates within them. And it's that rotation within there causing that shape change that makes the ATP to be synthesized. Now, somebody was worried that I said in class that this rotated counterclockwise, and somewhere in the notes it said it rotated clockwise. As I said, replied to the person, I said in class it really depends on which way you look at it. If you look at it from the top down, then it's counterclockwise. And if you look at it from the other side, it's going to look like it's clockwise. So the clockwise, counterclockwise really isn't important for our purposes. All right? But this guy here has got three sites. All right? And one of the sites is what I call open. And somebody also said that, well, in the highlights, I said this was binding ADP and PI, but in the class, I said it was releasing ATP. All right? Well, the primary function of the open site is to release ATP. Absolutely. All right? But as this thing is rotating, we can imagine we have transition. This is going to transition to L. And it's during that transition phase that the ADP and PI are binding. And that's what this is trying to show you. If you know it as ADP and PI are binding in L, I'm happy. Okay? Just understanding what the various roles of these components are. So the role here is releasing ATP. The role here is binding ADP and PI. And the role here is scrunching them together to make ATP. That's the three roles of those three complexes that are there. They move, as this guy moves around, O becomes L. L becomes T. T becomes O. And that makes sense because T going to O is going to happen because once you're at T, you've made ATP, you want to release it. And that's what the function of O is going to be. Does that make sense? So that's what's happening in this complex. And, you know, I've taught this class now for, 19 years, I guess. And I've yet to find a good figure that everybody likes about this. I don't know if it's just difficult to draw or conceptualize or what, but I've yet to find a figure that people were very easy to understand what's happening inside inside the complex. Yes, sir.
So how does the action of the proton moving down gradient actually cause the gamma protein?
Yeah, that's a biophysical question. So his question was, how does the movement of protons cause the gamma unit to move? I'll give you a simple answer to that, okay? But the answer really is much more of a biophysical one, okay? The simple answer is that protons are charged. And you can imagine charges moving in as they bind into they actually come in a little chamber. As they come into the chamber, they're going to cause, first of all, by the nature of their charge, an interaction. All right, so let's imagine that we have a positive charge that's sitting there to start with. All right, so that's adjacent. So I have a chamber. This comes in. I've got a positive charge over here. What's going to happen with two positive charges? I'm going to repel. Well, if this repulsion is part of the actual gamma complex itself, I've just moved it a slight distance. Well, I move it a slight distance, and now a carboxyl comes along that the proton can jump onto, and now the proton's on there. Or let's say an amine. It'll make it a positive. Put an amine on there. Right? Now I've got another positive charge, another one comes in, and so forth. And so it goes sequentially like that. All right? The remarkable thing is how rapidly it goes. And actually, I just remembered. Somebody asked me in class evidence for this, and I didn't tell them the evidence for this. There's a very cool video that proves that there actually is a rotation. All right? I'll tell you about this briefly. And what they did was they took a gamma complex and they freed it of this stuff, and they attached to it a long fiber of, I want to say it was myosin or something, some muscle protein, but it was a long fiber that they could actually see in a microscope. And then they fed the thing ATP. So instead of generating ATP, they fed it ATP, and they ran it backwards. But when they ran it backwards, of course, it was still rotating. And so by doing that, they were actually able to demonstrate that the rotation is a real-life phenomenon that's happening. So it is a real-life turbine that does it. And the rotation rate is quite rapid. Pretty cool. Yes? Don't worry about that. Don't worry about that. Yeah. Yeah. Yes, back in the back. Well, okay, so that's a good exam question, right? What do you think the other three subunits might be there for? Store ATP? Well, perhaps. Structure. It's absolutely structure. So, this is a complicated structure. We see it, in fact, if you look at the actual image that's out there of the structure, which I think I have somewhere here. ATP synthase structure. Okay, so now that's just a part of the structure. Blast it. I thought I had. Well, there's a 3D model of it, but we're talking about a lot of proteins that have to come together and do things. When we see protein, multi-protein structures, not every protein in that structure is performing a catalytic function or a movement function or something like that. A ribosome is a really good example. We'll talk about ribosomes next term. And ribosomes have like 50 proteins in them. And a good deal of the structure or the function of the individual proteins in the ribosome is simply to give it the shape that it needs in order to get everything into the right position for the formation of peptide bonds to occur. So that is actually what's happening here, that you have to position these guys in the right place, and that's what the other subunits are involved in doing. Yes?
I had a question when we were talking about
pain.
You mentioned the arachidonic acid
Yep.
Being converted into the, I can't remember what it's called.
So arachidonic acid gets converted into prostaglandins.
Right. And the prostaglandins synthesize the COX.
Yes?
So what is causing the actual transmission of pain?
Okay, so this is a common question. So how is it that prostaglandins cause pain, I think is your question. All right. So that's a little complicated, but if we think about pain that's happening, right, that if I poke my finger with a needle, what have I done to cause pain? So I had to have stimulated, in some sense, a nerve cell that is responsive to maybe puncture or maybe touch or something. We don't know an awful lot. In fact, of all the different nerve cells, touch is the one we know the least about. But I'll give you a good example. You have cells in your body, all over your skin, in fact, that respond to pressure. You push them, you can feel the pressure. Those are actually involved in touch. Okay? And pain receptors are very much the same as touch receptors. Okay? So if you look at what prostaglandins do, one of the things that they do is, and they do many things, but one of the things that they do is they stimulate swelling. Okay? And they can stimulate it pretty quickly. Well, imagine that you have swelling in a place where you don't normally have swelling. What are you going to do to nerve cells in that adjacent region? You may be stimulating them to fire, and that's one of the ways in which prostaglandins can stimulate something that ultimately results in pain. So the example I always give is a bee sting. Bee stings really hurt. You get right down to it. They really hurt, and they cause almost instantaneous swelling, and that's happening. It's pressuring a lot of different nerve cells. Okay? Yes, back here. Okay, so that's always a common question, so I'll be happy to say something about that. So if I go to my lecture here, and I talk about the SREBP pathway, this is the figure you're talking about right here, right? Okay, so let's go over the nomenclature I gave you first. All right, so if I talk about just the protein as the protein, I talk about SREBP. That's the thing. If I talk about it in the endoplasmic reticulum, I call it the SREBP-ER. If I talk about it in the Golgi apparatus, I talk about it the SREBP-G. And there are three parts to the SREBP protein. There's an R part, an M part, and a D part. Okay? Those three parts have different functions, as you can see. In this case, the R part stands for regulatory part. It's binding to this protein called SCAP that I didn't talk about in class. Okay? But it's regulated by that. It's held by that protein. All right? When cholesterol levels fall, this complex moves from the endoplasmic reticulum to the Golgi apparatus. And that's what's happening down below. Okay? So this is moved now to the Golgi. It's still in the membrane. It's in the Golgi apparatus. All right? But in the Golgi apparatus, there is, in fact, a serine protease that recognizes this whole thing that will cut that. Okay? So when it cuts that, you no longer have this being held to this, and the cutting of that is important because this has to be free in order to interact with the next protease. You might say, well, why doesn't this protease just come over and bind to it? Okay? Keep in mind that these are very simplistic diagrams. It won't have access to cutting here to cutting up here unless this has been freed of that. So once this gets freed of that by that serine protease, then the metalloprotease can clip here. So it clips and releases the D part away from the M part, and the D part then is able to go back to the nucleus where it binds to the steroid response element that is a DNA sequence inside of the chromosomes. The binding of the D segment of the SREBP to the SRE stimulates transcription of all genes that have an SRE. And one of those genes is HMG-CoA reductase, and that causes the enzyme to be made. So when the cholesterol levels are low, HMG-CoA reductase will be made. Okay?
Is there anything else that causes this to happen?
Is there anything else that causes this to happen besides low cholesterol? That's actually a very good question. And there are other things that will do that. That's correct. So we're looking at one of these. And the reason that this is called the steroid response element is that there are many different pathways involving steroids that can also activate this. All right? So we can imagine that there's a lot of different ways in which this enzyme might be turned on. This steroid response element, the SRE that's up there, we'll talk about eukaryotic gene expression control later in the term. Eukaryotic genes tend to have elements like that one element that's there that can activate it and affect it, but they usually have many elements that may have other effects on that. So even though this element might be being turned on by something, another element in front of the same gene might be turning it off. And so then it's a matter of which one's going to win. Okay? So it's very complicated in the case of eukaryotes, much more so than it is in the case of prokaryotes. You had a question here, then I'll come back here.
Is the serine protease not present in ER?
Is the serine protease is not active in the ER. It's only active down here in the Golgi apparatus.
I have another specific question regarding pumps. To be electrogenic, does the pump have to be pumping ions against the gradient, or can we go in?
Okay, so this question is basically what does electrogenic mean with respect to pumps? So electrogenic has nothing to do with something against or for. It has to do with a net change of charge as a result of the movement. So that's all it takes. So once you see the movement results in a change in charge, then you've got an electrogenic pump. Okay? Yes? Yes, we have. So, for example, the sodium glucose pump is electrogenic because it has sodium coming in, but nothing going out to balance it. All right. The lactose permease uses protons as a carrier for the lactose, and that's also electrogenic because there's nothing balancing that. So that's two that come to my mind. And I think that I talked about the calcium, briefly talked about the calcium ATPase, which is also electrogenic. So most of the ones I've talked about are actually electrogenic. Yes?
The figure that's on the screen, after the SRE is bound in the DNA,
Yep.
what's the length of the association for that? Does it disassociate spontaneously?
Very, very good question. So it's going to depend on the individual proteins and the affinity that they have for their DNA. So DNA binding proteins do vary in their affinity and also in the range of sequences that they will bind to. So it depends a lot on that sort of combination of factors. We think about, for example, I said, okay, SRE, this is a sequence that's in front of the DNA, and we think about, okay, well, that's very simple. It binds or it doesn't bind. But in fact, one gene might have an SRE sequence that's slightly different than another, that's slightly different than another. And so a binding of an SRE, of a D part of the SREBP may be much stronger for one sequence in front of one gene than it is in front of another. And that actually plays a role in determining how long it stays on, which is what you're asking, and how much RNA is made from it. So we'll talk about that when we talk about transcription. But very good question, yeah.
And one other thing about this complex,
Yeah.
after you've done the two different cuts and the metalloprotease and everything, is it that structure is scrapped, completely gone, or does it
Yeah.
regenerate?
So all proteins in the cell have a turnover. I haven't talked, it's unfortunate we don't have time in this class to talk about it. In fact, I skipped over the part about the degradation of HMG-CoA reductase. But all proteins in the cell have a turnover. And they have, the cells have a mechanism, it's called a proteasome, for breaking down things that they no longer need. So once things have been used and no longer are needed, they will ultimately be targeted for the proteasome for degradation and recycling of their amino acids. So that's ultimately going to happen to these guys. Yeah. Yes? Yes.
There are, I don't know, like there are kind of different types of pain.
Yep.
Are prostaglandins only involved in acute pain like you were describing, or would they also be involved in other types?
Yeah. Okay, so the question is, are prostaglandins only involved in acute pain of the type I described, or are they involved in other types of pain as well? Prostaglandins are very acute in the way that they work. Okay? Prostaglandins in the body turn out to be fairly unstable, and so they don't travel very far. And so as far as I know, and again, I'm not a medical doctor, but as far as I know, they're only involved in very localized pain because of the unstable nature of them.
Right, so it's chronic pain,
Chronic pain problems probably are not associated with prostaglandins, though when you think about some of the chronic pain like rheumatoid arthritis and things like that, where they're taking aspirin that are helping to relieve that pain, that would sound like there is a prostaglandin component to that. So, but again, it's localized. It's not something what I'm thinking of. I've got a pain all the way up and down my arm. It's localized to my joint. So localized pain will be prostaglandin. Yeah.
Are the prostaglandins ubiquitous throughout the body?
Yeah, are they ubiquitous throughout the body? The prostaglandins, it's interesting. You guys are really interested in prostaglandins. Prostaglandins, I'll talk a little bit more later about, But since you've asked the question, I'll answer it for you here. Prostaglandins, there are many prostaglandins, and they have really incredibly diverse set of roles. Some of them favor muscular contraction, like ones that are involved in uterine contraction. Others favor muscular relaxation. Okay? There are some that, in fact, one of the major roles of prostaglandins is in making blood platelets sticky. So you take aspirin as a blood thinner because you're inhibiting the synthesis of the prostaglandins that make the platelets thicker. That's what the blood thinners actually are working on that basis. I talked in class about prostaglandins that are involved in stimulating the growth of intestinal cells. There's all kinds of roles prostaglandins have. But they're all fairly unstable, and they don't travel very far. That's one thing about them.
So an NSAID would include all prostaglandins or just specific prostaglandins?
Yeah, so an NSAID, his question is, will an NSAID inhibit the synthesis of all prostaglandins? It's a complicated question because it turns out that there's more than one COX enzyme that makes prostaglandins. So we don't fully know the story of that. We sought that, as I think I mentioned in class, with making the inhibitors to the COX-2 enzymes. And those made a specific group of prostaglandins that were very localized to joints. Okay, so there's a localized pain for you right there. And they thought they had that all sorted out, but then they discovered that some of these same ones were being used in the heart and causing problems. So it's a complicated story with prostaglandins. Yes?
All the way back to the question about the mitochondrial outer membrane.
Yeah.
How we shouldn't assume that the pumped protons will just stay there because they can go through the outer membrane.
Okay.
Does that mean that really metabolically active cells that have a lot of mitochondria have to have protection for stuff that happens outside because there might be a buildup of
Okay,
mitochondria?
so our question has to do with the mitochondria of very active cells. And are their outer membranes different than those that are of mitochondria that aren't very active? Is that the question?
Well, I guess I didn't phrase it very well, but more like an allowance for the escaping protons being of a higher concentration and something to mitigate that.
Okay, so a question has to do with actively metabolizing cells and the concern about maintaining the gradient of protons for those rapidly metabolizing cells. Is something done to constrain those so that you favor more the synthesis of ATP? Is that safe to say?
Yeah, and just the controlling how acidic your external environment is.
Okay, so to answer your question, what I would say is rapidly metabolizing cells have a couple things. One is they make more mitochondria. That's reflecting the fact they have higher needs of ATP. And I think you can make a pretty good argument that if they're rapidly metabolizing, they won't actually make gigantic proton gradients. Because once the proton gradient starts to form, it's going to, that system is actively using things and bringing protons back in. So I'm not an expert in this field. I say that a lot, but I'm not an expert in this field. But my strong feeling would be that that would not be a factor. Now, you do see some things with actively metabolizing cells that you don't see with other cells, and that is you see a lot more damage to mitochondria. And that's because you're making reactive oxygen species in the process of going through all of these things. Okay, so all right, so that's a consideration that you see in rapidly metabolizing cells. Yes, Eric. Uh-huh. How does this guy move from here down to here? Okay, keep in mind that based on that figure I showed you last term, that Golgi apparatus basically is a budding off of the endoplasmic reticulum. So that's really the mechanism by which that's happening. Yep. Yep.
Can superoxide dismutase be given to people who smoke, who have the reactive oxygen?
Okay, so good question here. So can you take a smoker and give them superoxide dismutase and mitigate the effects of reactive oxygen species in those smokers? I would imagine that would be difficult to do. And the reason it would be difficult to do is that the way we take things in is through our mouth and into our stomach, which is going to denature almost any enzyme we hit it with. Can that be done intravenously? I imagine it could. I don't know if anybody's done that. And I think most people are very nervous about doing things intravenously. for a couple of reasons. One is you may stimulate an immune response, which would be a very bad thing. And second, we tend to think of things as good or bad. This is a bad thing to have this reactive oxygen species here. I was talking to a student earlier today about hydrogen peroxide. Hydrogen peroxide is a reactive oxygen species. And so he had a very cool plan for stopping the production of reactive oxygen species. It was interesting. I mean, it was odd, but it was interesting, okay? I like the plan, all right? But the problem, as I told him, is that hydrogen peroxide is actually used inside of a cell to signal. So if you stop the production of hydrogen peroxide, what would be the effect of not having that signal? There might be some bigger problems with that. So I think it'd be similar to what you would worry about here. Yeah. Yes, back there. Yeah, sure. There's a very simple difference between those. So his question has to do with the difference between the P-type ATP transporters and the ABC cassette. The real differences that I've emphasized in class on these are that they are simply due to structural considerations, okay? Simply due to structural considerations. If we look at the sodium-potassium ATPase. Sodium-potassium ATPase is an example of a P-type transporter. And P-type transporters will always have an intermediate in them, a covalent link between phosphate and aspartic acid, forming something called a phosphoaspartate. ABC transporters will not have that. They will not go through that covalent intermediate. So it's like the difference that we saw between the serine proteases, which formed a covalent intermediate with the serine during their reactive cycle, and that had to be resolved. The same things happen with P-type ATP transport proteins. The ABC mechanisms are more like the spartyl proteases, which didn't have a covalent intermediate because there was nothing to hold those together. And in this case, they're simply using the hydrolysis of ATP as the driving mechanism. There's structural differences, and that's beyond where we're going to talk about here. But that's the gist of the differences between them. Make sense? Yes?
Could you briefly go over the photosynthetic fish idea with the bacterial rhodopsin?
Photosynthetic fish with a bacterial rhodopsin. Yes, one of my favorite topics. I love that. Okay, I see a lot of smiles. People must also like that. The photosynthetic fish is a very simple idea, and the simple idea is to simply use light as a way of making a proton gradient. All right. So when we look at what plants are doing, for example, just to give you some background, we don't talk about photosynthesis in this class, amazingly enough. When we look at what plants are doing, they work and they make proton gradients, okay? So they work very much like what the mitochondrion does. The odd wrinkle about plants is that instead of pumping protons out of a chamber, they pump them in. So they create sort of a reverse proton gradient compared to a mitochondrion. They're pumping protons into the chloroplast. It's still a proton gradient, though. And they use an ATP synthase that's very much like the ATP synthase that is used in the mitochondria. In fact, it's virtually identical in the way it looks. Okay? So my idea was, well, if you can construct a proton gradient, if you can use a protein that is able to pump protons solely using light, then you can make a mitochondrial gradient that you want without using oxidation as a means of moving the protons. So that was my idea, and it still is my idea, I guess. The idea being that you put this light-sensitive protein into the inner membrane of the mitochondrion. And the reason I picked a transparent fish was so I could get more light all the way through the body and get as many cells exposed as I could. You shine the light, the proton pump kicks the protons out of the mitochondrion. You have to position it right, so it kicks them out of the mitochondrion, creating a gradient, and then that gradient is used to make ATP. Well, I didn't have to eat anything to make that happen. I didn't have to oxidize anything. I make an ATP, excuse me, simply on the basis of shining light on that organism. Now, as I said, you would still have those organisms would still have to have a carbon source. They've still got to make things. Plants don't have to have that carbon source because their carbon source is carbon dioxide. They can pull carbon dioxide out of the air. That's what true photosynthesis fully is. In photosynthesis, there's a light cycle and a dark cycle. the light cycle involving the proton pumping, the dark cycle involving pulling the carbon dioxide out of the atmosphere. So we'd be making as a fish that has a light cycle but not a dark cycle. Makes sense? Somebody make this damn fish. I want to see this. Yes?
Can you talk about the Melee shuttle?
The Melee shuttle, the Melee aspartate shuttle, sure. So the Melee aspartate shuttle is an important shuttle, and the reason it's important is because the NADH does not cross the mitochondrial inner membrane. So if we want, got the wrong thing. If we want the electrons from the cytoplasm, and the cytoplasm, there's a lot of reactions that will make NADH. You've seen glycolysis, and glycolysis produces NADH. If you don't do something with those protons, then the cytoplasm is going to be stuck doing fermentation. So it's going to have, if the cytoplasm can't convert NADH into NAD, right, then pyruvate's going to make it to lactate, and when pyruvate makes it to lactate, lactate isn't going to make it into the mitochondrion, so you stop everything there. So getting these protons in from the cytoplasm into the mitochondrial matrix excuse me, is very important. It's a very important thing because there's a limited supply of NADH and NAD out here. So this shuttle takes on a lot of importance in terms of keeping everything going. So the shuttle works by starting with oxaloacetate, which is abundant out in the cytoplasm, and it combines or takes the electrons and protons from NADH and makes malate. Malate is moved across the mitochondrial inner membrane by a transport protein that is an antiport for malate and alpha-ketoglutarate. All right? As I said in class, okay, that's fine. Not a biggie, right? The important thing is we're moving electrons in. Here's these electrons coming in on the backs of malate. And when it gets inside the mitochondria, exactly the opposite reaction happens. Malate dumps its electrons and proton onto NADH and becomes oxaloacetate. Okay? That's what's happening. So the critical part of that whole thing is right here. This is the most important thing that's happened in this process. Everything else is simply to balance the equation. That's all that everything else is there about. Right? Yes?
So does this precede the citric acid cycle? Like, or does this bring in the ingredients of the citric acid cycle to happen within the matrix, or does the citric acid cycle already happen and create it?
I'm not sure I understand the question fully, but the citric acid cycle, remember that this is not, it's tempting to think of this as this happens, then this happens, then this happens, then this happens. But this is going on as the citric acid cycle is going on. So it's bringing in NADH, right? Or it's bringing in electrons and making NADH. What's that going to do to the citric acid cycle? No, it's going to slow it down, right? So this is where the amount of oxygen that's available is going to be critical. So because both things are sucking, all right, the NAD and making NADH. The more NADH we have, the slower the citric acid cycle is going to run. So oxygen becomes very critical at this point. This is why oxygen moving across membranes all by itself is really cool and important. We don't need a transport mechanism to move oxygen. We don't need a transport mechanism to move water, carbon dioxide, and carbon monoxide, all right, because they can freely move across there. But getting enough oxygen in here to keep the electron transport system going is going to be critical to bringing this guy down to NAD. Make sense? Yes, Omar? Yes. Yep. What about it? What's the question? Oh, can I go over it? Okay. Well, it's actually much simpler by comparison. If we look at what happens in that shuttle, we can see that it works by reducing dihydroxyacetone to glycerol 3-phosphate. And this glycerol 3-phosphate is handled by this mitochondrial enzyme, the dehydrogenase, that passes things off to the FADH2 that's in complex 2. This is sort of a simplification of that process. But it's basically a 1-2 step process. Right? Nothing has to move across the membrane. Nothing's moving across the membrane. There's no transporter proteins moving things across. This is dumping things right straight into this complex, which is itself going to feed into complex 2, into the electron transport system. Okay? Okay, Q cycle. What questions did you have about the Q cycle? What would you, okay, so, all right, so, as I say, I give lectures on these, so I want you to review those lectures, but I'll say something about it here. Q cycle looks really complicated, and Q cycle is really simple. I mean, it's really, really simple, all right? Complex 3 is a docking station for everything, okay? It's a docking station for coenzyme Qs called QH2 that are coming in from the pool, which are just things that are mixed around the membrane. It's a docking station for cytochrome C. It's a docking station for partially reduced coenzyme Q. That's all it is. So the Q pool lets in QH2, right? QH2 has the electrons. It's in the reduced form. That's why we call it QH2, meaning it's got 2 protons, 2 electrons to do that. So the 2 electrons split. One goes to cytochrome C. One goes to a coenzyme Q that has no electrons. Now it's got 1 electron. It goes from a charge of 0 to a charge of -1. Okay? This guy's reactive as hell. It can't be released freely. If it were, it would just go out and react with everything. So it's contained within this docking station of the complex 3. QH2 has dumped off its electrons. It dumps off its protons, which get pumped, and the Q is released. So we've just basically docked, unloaded, and emptied, and now replaced the ship. So we have a new ship coming in, and the new ship comes in, brings in 2 more electrons. 1 electron goes exactly as before to cytochrome C. 1 electron goes to the other coenzyme Q. All right? And by this point, it has 2 negative charges. It picks up 2 protons, and now it goes back out to the Q pool. Right? So, but the same thing happens. 1 electron up here, 1 electron down here. That's all there is to the Q cycle. Yes?
For the two protons that the Q with a negative sign, that will fix an electron in those 2 protons. Those 2 protons are from the QH2 or they're just from like outside?
I'm not sure I understand the question, Omar.
The 2H plus, that's
So these are coming from the matrix.
okay. So
Yeah.
like we're pumping it. So like pumping.
This isn't pumping. I mean, they're just simply attracted here. The pumping is going on when they're kicked out of the mitochondria. That's pumping right there.
Okay, so we're pumping it outside of the matrix. But those protons are in the matrix.
These protons are coming from in the matrix, but they're just attracted to this negative charge. So this is not a pumping. There's no gradient because we haven't crossed a membrane of any sort. Okay. Does it make sense? Okay. Yes.
I don't get why that QH2, when you ejects it out of the matrix, why wouldn't you just give it to the Q minus down there, 2 H's? Why do those 2 H's have to come from the matrix?
Why doesn't it come from where?
Why do those 2 H's have to go to the Q minus? Why can't those 2 hydrogens instead come from the QH2 at the center?
They're pumped out here.
Yeah, it just seems like it would be easier for them just to go straight to the Q.
Well, again, this is a schematic representation of what's going on. If you think about what's the aim of this whole process, so I'm going to try to answer your question another way, is making a proton gradient. So if I'm simply shuffling protons back and forth here, I'm not accomplishing some of the things I want to do, right? But what you said is right. I mean, there's no, if I did that here, okay, then there's no net gain of protons out, right? But I do have protons out here, right? So in essence, that could happen, okay? But, you know, you're very tempted, and people, it's very tempting because we talk about this as this goes to this, goes to this, goes to this, and we see this proton, we see this proton, we see this proton, and we see this proton. But, you know, we're talking about things that are just getting picked up out of the solution, and there's millions of these that are there. Absolutely millions of them that are there. Okay? Yes?
Just for clarification, from what I see there, it looks like Q and QH2 both are part of the pool. Is that correct?
That's correct. That's correct.
And those are both coming from the inner, that looks to me.
So the Q pool is, so keep in mind that coenzyme Q is a molecule that's embedded in the mitochondrial inner membrane. It's embedded in there. So it freely floats around. Because when we drew the scheme originally, we said that it was carrying electrons from complex 1 or complex 2 to complex 3. So it's just diffusing through that membrane is what it's doing. So we just use the term pool to describe all of those. That's all that we're doing with that. All right? When something in that membrane bumps into complex 3, this is what we get. So one of the things I think it's really hard to grasp, and it's remarkable, is to think that all of this is diffusion. This is all diffusion driven. Okay? In this case, we have diffusion that's happening within the intermitochondrial membrane. But nonetheless, it's simply diffusion that's driving the whole process. Yes?
So will the Q and the Q pool eventually become QH2, or will it be used as Q somewhere else?
Will what now?
The Q that exits the complex and goes to the Q pool,
Yeah.
will it become QH2 in the Q pool, or will it be...
Okay. So the question is, what will happen to Q once it goes into the Q pool? Well, keep in mind, what it's done is it's dumped its electrons off. So it's got to go back and get more. So where does it get more? Complex 1 and complex 2. Right? So it's just going to go back and forth and back and forth. That's all it's going to do with all these. I like to think of Q and also cytochrome C as a shuttle. Grab electrons here, carry them over here. Grab electrons here, carry them over here. Cytochrome C takes them from 3 to 4, 3 to 4. So they're just shuttles, shuttling things back and forth. And that's because they're smaller than these complexes. They're very large. These complexes don't diffuse very rapidly. They don't move very fast in the membrane. So these smaller molecules like Q and cytochrome C can move very rapidly, and therefore they're better for transporting those electrons. Yes, I'm sorry.
This might be really simple, but when you said the 2 carbons added from acetyl CoA in the beginning cycle do not become oxide to carbon dioxide beginning the second time, like so you don't lose, so you go around once and there's no carbon. I was just
Oh, and you're talking about the citric acid cycle?
yeah.
Okay. So, no, I think you are confused. Let me just show you what I'm talking about. So if I go to the citric acid cycle, and, nope, that's the wrong one. Yeah. So here's the thing I think you're referring to. So we see 2 carbons coming in. We see 2 carbons exiting, but the 2 carbons that came in aren't the same ones that exit. But you still have 2 carbons exiting because otherwise we would not have the oxidative decarboxylation, right? So you see what's happening. The CO2 that's coming off is black, meaning it was one of the original black ones here. But now, after this has gone around one time, now we color them black, but in essence, there are green guys that are in there from before, right? So now the greens have become black, and they'll be lost on subsequent cycles.
So the first time it's the pre-existing carbon, and the second time it's one from the new one.
That's correct.
Okay, thank you.
Good. Yes, Eric.
You talked about cholesterol synthesis.
Yep.
So you're talking about cholesterol being running into external storage.
Yes.
Is there a preference between one?
Yeah, boy. So that's a very good question, actually. So cholesterol in the body, I mentioned it in the highlights. I didn't mention it in class, but since I did mention it in the highlights, I'll say it here. Cholesterol in the body comes from 3 things. Okay. So it comes from synthesis, from diet, and from storage. All right. And the dynamics between those is a little hard to sort out because how do you distinguish dietary cholesterol from stored cholesterol, for example? Okay. Synthesis may be a little bit different, but dietary versus stored is going to be a little hard to sort out. Is there a preference for one versus the other? I guess it depends on what you mean by preference. So, cells that need cholesterol, if nothing else is available, they will make it. And if there's abundant cholesterol floating around outside them and they don't have to make it because that's energetically intensive, then my gut feeling is that would be a preferred way to go. But you also have to think about the entry of cholesterol into a cell is also kind of, what's the word I want? Invasive, as it were. Okay, so it has to be internalized. Those LDLs have to be internalized. That may have some costs associated with it as well. So it's hard to answer the question in terms of what you mean, what preference really is. Okay, but the energy consideration is probably the one that drives everything. Yes.
Could you go over the desolvation, resolvation energy with respect to those?
Okay. So, anything specific?
Well, I was looking at that slide with the arrows. Can you just quickly explain that one slide?
Okay. So, let's see. That was here, I think. Okay. That wasn't the one I talked about in class. The one I talked about in class was all right. I'm not sure what figure I had there. Oh, this is the figure I talked about in class. Okay. All right. So the problem arises from the fact that you want to have selectivity for a specific ion, but you've got some ions that are bigger in diameter than others. So potassium is bigger in diameter than sodium, and consequently, if the only basis you have to exclude them is the diameter of that, then you would say that there would not be any potassium selective channels. Right? Well, it turns out that there are potassium selective channels, and it means that because they're potassium selective channels that don't allow much sodium, a little bit of sodium gets in, but that don't allow much sodium to get in, that there must be something besides the basis of size for excluding the sodium. And it turns out that there is a difference, or there is a basis of selection between the two, and it relates to what's called the energy of solvation. And the energy of solvation is due to the fact that these channels that these guys are passing through are tiny. So even for something that is a sodium, for example, it's going to, as it tries to pass through the channel, it's going to lose waters that are attracted to it. And any ion in solution is going to have waters attracted to it because waters have partial negative and partial positive charges. So you've got to strip those off. And the stripping off of water really comes at the cost of energy. There's an energy investment that has to happen with that. Well, you'll notice that we're not using any ATP in this process. So the overall energy process has to be reasonably favorable for this to happen. We're not pumping anything. These guys are coming through on the basis of diffusion. This is happening in a nerve cell, for example. Okay? So the energy considerations then have to be different for a potassium moving through than they are for a sodium moving through. And they are different. And they do relate to the solvation energy. Okay? So if we look at what happens with the potassium ion, there's a certain amount of energy that's required to be put in. That's the upwards arrow. That's required to strip those waters off of the potassium. That potassium has a shape and a dimension such that desolvation is energetically not as costly as it is to do it for a sodium, because things are positioned exactly around for the diameter of a potassium ion, and they're not situated for that for a sodium. So the sodium, actually, because it's smaller, has to bounce around some in there, all right, before the waters get pulled off. That takes energy. Okay, so it takes more energy to pull off those waters from a sodium than it does off of a potassium. So it starts out being more energetically costly to begin with. Then as this guy is moving through the chamber, okay, again, all the different things are there. The charges are there. The geometry is there to work with the diameter of a potassium ion. And it's optimized so that when it comes through and it gets to the other side, it's gaining those waters in a manner that's very efficient. And energy is released. So you can see that there's energy put in, there's energy released. In this case, there's more energy released than put in. So it's favorable for this guy to pass through. Over here, the geometries are such that it doesn't fit in very well. So sodium, again, is bouncing around inside of there, and it's not efficiently and effectively picking up waters as it's making its movement through that chamber. That means, therefore, that there's not going to be as much energy released in the resolvation of sodium, and this becomes energetically unfavorable. Now, this will still allow some sodium in. there's still a little bit of sodium that's going to make it through, but not nearly as much as if it were simply done on the basis of size. Okay, hopefully. Yes?
Two questions about this. First, is there a channel length component to this? Would it, if you made the channel longer, would it be more selective? Maybe get less potassium through?
You know, that's a very good question. I think you could make an argument for that, yes.
And then secondarily, with the way the geometry is inside.
Let me back up, and another thought just occurred to me. So the longer the channel's going to be, the slower things are going to move. And you would worry, I guess I would worry about plugging over time, right? So when we think about this happening in a nerve cell, for example, speed is of the essence. So we sacrifice something in terms of accuracy. And there's not much sacrifice. To give you an idea, I think it's about 1,000 to 1 that we see preference of, no, it's maybe 10,000 to 1, preference of the sodium channel blocking potassium. It's about 100 to 1 for the potassium blocking sodium. Okay, so it's still very efficient, but there's a price that I think that you're paying in terms of the speed of making this happen.
Do you speed up your nerve signaling by shortening the channel? Do you have quicker reaction?
Well, again, I think going back to your original point, if you shorten it, you probably would have less selectivity. Yeah. So, and that selectivity is important because it's these waves of ions that are moving through here that are considerations in nerve transmission.
And then the second question I had about the channels was about the internal geometry. Is it kind of tumbling, or does it get set and kind of tracks and go through?
Yeah, that's why this other figure, which I should have showed to class, and I didn't. I should have showed this in class. I think actually helps you to see that a little better. And that's right here. Okay. So we can see that here are some potassium ions coming in. They bounce around to get to their location. And here's one that's gotten stripped off of its water. All right. The movement of one through literally starts pulling others through with it. So it's a pretty active process that this is happening and going through there. But again, it's diffusion driven.
Okay, but it's not like a twisting thing where the potassium tumbles over itself. The orientation.
I would say no.
Okay.
Yeah. But I'm not a biophysicist either. So, yes, sir. Okay.
For digitoxin,
Yes?
how is knocking out the potassium and sodium pump making calcium pump less likely to work?
Okay. So the question was, how does digitoxygenin work, basically? Is that the question? Okay. So digitoxygenin, remember, first of all, that it blocks the sodium-potassium ATPase. All right? So the sodium-potassium ATPase is necessary for pumping sodium out and potassium in. So that creates a sodium gradient. It also creates a potassium gradient, right? In the case of sodium gradient, greater concentration of sodium outside than inside. In the case of potassium gradient, greater concentration of potassium inside than outside. Well, the sodium gradient is relevant because the sodium gradient, there's another electrogenic pump that somebody asked about earlier. Okay. The sodium gradient is relevant because the sodium gradient is used to pump calcium out of cells. So the gradient of sodium wanting to flow back in is used as an energy source to pump potassium out. Right? If you have a sodium gradient, you will pump potassium out. Well, not potassium, it's calcium. I'm saying potassium. Calcium, pumping calcium out. Right? All right? So if you are pumping calcium out, and it's a nerve cell that you're pumping it out of, calcium is necessary for muscular contraction. So if you pump calcium out, you're going to have less strenuous muscular contraction.
I thought the cell had the calcium ATP.
Okay, so his question is, the cell has a calcium ATPase. So what about that? Okay. All right. So there's many pumps. All right. So it's hard to look at them in isolation, I understand. But for the moment, that's what we have to do. The calcium ATPase is used to sequester calcium inside of organelles. Okay? So when we talked about the sarcoplasmic reticulum and the endoplasmic reticulum and how calcium can't be in high concentrations around the nucleus because it'll precipitate the DNA, this calcium ATPase is used as a sequestration mechanism for that calcium. So that's doing something different than what this is doing. So in muscular contraction, the release of calcium stimulates muscles to contract, period. All right? So if we reduce the concentration of calcium inside of muscle cells, they will not contract as hard. Therefore, if we have a lowered sodium gradient, we will pump less calcium out, and the muscle will contract more. If that's a heart cell, then you've just made a stronger heartbeat. Okay? Now, digitoxygenin is a poison. Okay? So we're talking about things that are used in tiny amounts, in targeted amounts, all right, to keep that calcium concentration in the heart cell low. All right? But it's important to remember that this is a poison. So that's why foxglove, if you try to eat foxglove, you will die because too much is going to kill you. Yes?
That sequestration of calcium, that effect, does that also help to draw more calcium into the cell overall because you're creating an effective gradient by sequestering some away?
Okay, so our question is, does the sequestration that's done with the calcium cause there to be a calcium gradient relative to the cell and thus favor the movement of calcium in? I don't know what the concentration of calcium is typically outside of cells. I can't answer that question. A calcium gradient would only be important as it would be used for something. Remember, so it would still have to go through some sort of transport mechanism to get in to do that. So I would say it's probably a limited importance, to be honest. Yeah.
So after it's pumped out, what happens to those cells?
They can come back in. They can get back in. Yeah. Okay. But during that time that they've been pumped out, that muscle cell is basically, it's important also in the cycle for release of the muscle cell. Right? We don't want it to just hold. We want a muscle cell to release, especially if it's a heart cell, right? They're doing this, right? So we want it to be able to release as well. Yes? Yep. So that's the relaxation. So we think about a muscle cell has to relax as well as to contract. So getting rid of calcium will be necessary for part of that relaxation. Okay? So with digitoxin, they're not going to relax as much, and they're going to stay contracted, and they're going to actually, that's going to result in what we perceive as a stronger heartbeat. These are given for people who have congestive heart failure. And when congestive heart failure occurs, the heart is not beating strong enough to pump the fluid out in the various parts of the body. So you can get accumulation of fluid as a result of that. Okay? Yeah?
I don't remember the details specifically, but I seem to recall that calcium was involved in blood clotting, so I was wondering what the effect of digitoxin would have on that.
Oh, okay. So calcium has a role in blood clotting, and so your question is, what effect would that have? My suspicion is the effect would be pretty minimal. It might have a slight effect, but pretty minimal because we're also talking about pretty small concentrations in a pretty small targeted area of calcium. So I don't know that increasing calcium at the levels that we're talking about here would have any significant impact on blood clotting. Remember that the blood clotting requires a cascade of events to happen, so it's not just the calcium that's there. It might help to make a clot better once the clotting process has started. I don't think calcium by itself would favor clotting. No. Okay? Good. You guys are thinking about stuff. This is great. I should have made the exam a lot harder. No? All right. Well, good luck. The exam is all done and ready, so it's ready for you tomorrow.